dear guests, it is a very special honor to me to introduce Professor Sakmann. This is really very special because 30 years ago, in 1983, as a young medical doctor doing research in the Krannert Institute of Cardiology, Indianapolis, United States, studying the cellular mechanism of antiarrhythmic drugs. In the lab, somebody came into the lab with a paper in his hand and showed this figure and told, it is not fantastic, it seems possible that now we can measure in the future the currents and the activity of a single sodium channel. And this was one of the first papers on the cardiac cell which showed the current flowing through a single channel. And this work was based after the inspiration of the work made Professor Sakmann and Erwin Neher, who a couple of years ago developed this very neat technique, and they could record currents through a single ionic channel. And indeed, it was fantastic, and it was a breakthrough and opened a new horizon in all electrophysiology, including my field in the cardiology. And Bert Sakmann and Erwin Neher were the scientists who did that. And they were awarded the Nobel Prize in 1991 for this discovery. I just here selected some of the major publications related to this discovery, and I highlighted two of them. One, I think, was the first one in Nature when this was described, and the other one is the Flügel's paper, which was cited more than 16,000 times, which is really a very high number, as was noticed by Professor Sabo earlier. Of course, there were many other publications. So since there are many students here who are not electrophysiologists and also scientists, they have research area other than electrophysiology, I just very briefly show the principle of this very important discovery. With a fine tip microelectrode, we push the cell membrane and make a tight seal, a giga seal, which prevent current flowing between the electrode and the cell membrane. So it is so the current which with amplifier it's possible to measure the current flowing through the single channel proteins, and the voltage can be controlled by different electrode or by different ways also through the pipette. So here the other side of the slide shows very similarly the same. If we make the suction even more, we can rupture the patch, and then we can measure the activity, all the channels in the surface membrane, and can measure the whole cell current with this technique very accurately and very precisely, comparing the previous techniques. And if we apply a different way the patch clamp technique, we can gently pull back the pipette from the cell, and we can tear down a little patch of the membrane. And this is the so-called outside-out formation of the patch clamp technique, and can measure the channels which is within the area which is covered by the pipette, and we can measure single channel activity, one or two. In this case, there is two, so two distinct channel opening. And this is really very important and very widespread technique nowadays. So Bert Sakmann, Professor Sakmann, was born in Stuttgart, 1942. He had the Abitur at the Wegenburg Gymnasium. During his years, he was, in addition to be interested in biology, also with technical things, started the medical school in the University of Tübingen and finished the medical studies in Munich. He started his career in Munich with Otto Detlef Kreutzwert. He was a very well-known, well-established physiologist. at the Max Planck Institute, and then he left for postdoc position to London and worked together with Bernard Katz in the University College of London, who won the Nobel Prize in 1970. So, he came back to Professor Kreuzfeld's lab. Professor Kreuzfeld moved to Göttingen, so now in Göttingen at the Max Planck Institute, and started working together with Erwin Neher until 1990. So then he took the position of head of the Department of the Cell Physiology in the Max Planck Institute in Heidelberg. And as I mentioned, 1991, he was awarded with the Nobel Prize shared with Erwin Neher. So from 2009 to 2012, he got the directorship of the Max Planck Institute in Florida, USA, and also since 2007 until present, he is the head of Emeritus Research Group of Neurobiology at the Max Planck Institute and Research Fellow for Neuroscience at the Technische Universität in Munich. Professor Bert Sakmann's recent interest is still in neurobiology, elucidating the three-dimensional structural arrangement in the brain, playing key role in memory, attention, thought, and also aiming to understand the pathophysiology of Alzheimer's disease. And please, Professor Sakmann, deliver your lecture.
So thank you very much for your very kind words, for your introduction. You actually have explained a quarter of my lecture, so I will be very short on that. And I would like to say that I come back with best memories to Szeged and to Hungary. And there are two occasions where young Germans admire the Hungarians. One is shown here. This was apparently, and I got this from the rector of the university, the first declaration in '56, where the revolution, the Hungarian Revolution, started. I was 14 at that time, and I could not be pulled away from the radio. We were listening all the time what's happening. And this is why I like to come back to Szeged to see where all this has happened. The other occasion, obviously, happened in '89, when again the Hungarians changed German history by cutting the Iron Curtain. This is maybe not so well known in other parts of Europe, but in Germany, Hungarians are very well known for these two events. There's another event which I won't mention. But I would like to stay with this and say I'm very proud to even meet Tamar Kiss, who is apparently one of the creators of this document. So what I'm going to talk about is from single channel currents to cortical circuits, which describes more or less how I began in neuroscience and how I'm going to end in neuroscience. Initially, I wanted to talk only about cortical circuits, but then Peter Hegge said it would be nice to spend the whole career to show students how interests change during a scientific career. So I'm going to talk first about the single channel currents, and then I'll switch in the second part to the analysis of cortical circuits, which I spent my time over the past 6 or 7 years. Now, this is a record. So what you see here is current recording through a muscle membrane, and what you see here with these short blips are the effects of the opening of individual ion channel. When an ion channel opens, a brief current is flowing, and this brief current is reflecting the opening and closing of an ion channel. You can see very clearly that there are two remarkable features. One of them, it has a unitary amplitude. These current amplitudes are unitary, meaning that the ion channel is either fully closed or it's fully open. You also can see that they have variable durations, and that's another feature of a simple chemical reaction. If you have a bimolecular reaction or even a monomolecular reaction, you expect that the lifetime of one of these measurable states is distributed exponentially, and that's exactly what we find. Now, due to this, to these single-channel currents, I was awarded half a Nobel Prize in 1991 for discoveries concerning the function of single ion channels in cells. And I shared the Nobel Prize with Erwin Neher. This is a recent figure of Erwin Neher. And we were awarded the Nobel Prize for discoveries on ion channels, and I want to explain what are these discoveries. These discoveries are, it's the first observation in real time of a single molecule in biology. It's the first time one could watch the transitions of a single molecule. Equally important, it is the first proof of the ion channel concept. The ion channel concept means that electrical signals by which our brain, our muscles work are due to the current flow across membranes via ion channels as opposed to carrier-mediated currents. Carrier-mediated currents also exist, but for fast signal transmission, which is required to react to our environment, the body is using ion channels. And finally, we were the first to identify structural determinants of the ion channel function. I'll briefly explain now what I mean by this. First, we had to develop a technique to measure ion channel currents, and this is a group of 5 people. This is Erwin Neher, Fred Sigworth, Alain Marty, myself, and Owen Hamill. I had to sit down in order to fit onto this photograph. So these were the people, or our collaborators, with whom we developed this technique. Now, what is a technique? You just heard about it. It consists very briefly to isolate a patch of membrane via the tip of a glass pipette that prevents the current flow between the inside of the pipette and the outside solution. So all the currents that are measured are flowing through these ion channel proteins, and this allows us to study their properties, which we just saw. They are either open or either completely open or either completely closed, and they reflect an interaction, in this case, between a signaling molecule called acetylcholine and the acetylcholine receptor. What you see with this arrangement is shown here. You have seen this from an original experiment, the very first slide. This is a current trace, and it switches between 2 states. The structural interpretation is that during this time, the channel is completely closed. At this time, lasting about 4 milliseconds. The channel is in the completely open state. It passes a current of about 4 picoamps in the time order of a few milliseconds. As I said, the time is not fixed, but it varies exponentially into a first degree. Now, to make this clear, what I said is the structural interpretation of this current trace is shown here. The ion channel, we have isolated the single channel protein. The channel is closed. However, if a signaling molecule, acetylcholine, which you see here as these green dots, attaches to the binding site, the channel flips into the open state, and ions, in this case sodium ions, will pass through the channel as long as the channel is open, and this is this current transition. So this is the measurement, and that's the structural interpretation. When the channel, when the acetylcholine molecule dissociates again from the binding site, the channel flips into the closed state, and as a current, we see an upward deflection. There is no more current flowing through the membrane. Now, the standard size of these ion channel currents in most tissues, and not only for acetylcholine receptor mediated currents, but let's say for sodium channels, potassium channels, chloride channels, with the exception perhaps of calcium channels, is in the order of picoamps. And what is a picoamp? In the household, you have to run your household gear. You need currents in the range of a few of amperes to 100 milliamperes. 1,000 is a milliampere. This is what you see in electronic circuits, like a PC or part of a PC. If you decrease the size again by 1,000, you get into microamps. The microamp is the current that flows through a muscle membrane when we twitch or when we move our muscles. Again, 1,000th of this microamp is a nanoamp. This is what you see in some biological membranes, like the end plate. And again, 1,000th, that is a millionth of an ampere, is the size of a current by which our nerve cells and muscle cells are operating. So these signals are generated by the superposition of these elementary currents. Okay. Somehow, can we turn down the light a bit, because I can hardly see. So this gives you an idea what a picoamp is, and the signals that by which our brain or our body operates is due to the superposition of between 10 and 1,000 of such elementary events. Now, these experiments, which finally resulted in the patch clamp technique and the accurate description of ion channels, is not coming out of thin air. We had a long period to develop, and we are very grateful, or we were building on the work of Sir Bernard Katz, who won a Nobel Prize in 1971. And he had discovered, together with Riccardo Miledi, that when you apply acetylcholine to an end plate membrane, you see an increase in noise. So this is a conventional current measurement, which we just concentrate on this. That's the background noise of your measurement. You apply acetylcholine and you get additional fluctuations. And using what's called a spectral or Fourier analysis, one can derive, using a few assumptions, the size of the elementary contribution to this noise. Now it happened that the estimate of the elementary current was about 1,000th of what one could develop, what one could measure with conventional techniques, but it was in a reach where we thought we are going to attempt it, because increasing the sensitivity by a factor of 1,000 didn't seem to be completely out of question. So this is really the basis of current measurements. These are very early experiments, and you see the experimental arrangement. This is a single muscle fiber. It's resting on a hook to mechanically stabilize it. This is the end plate that contains these receptors, which you have seen, and it contains a small amount of acetylcholine, and this is the patch of membrane that we are isolating. The first recordings were just so good that we thought it's worth to improve, to work on this. So this is 10 picoamps, and we cannot resolve very nicely individual single channel currents, but it was clear that if we increase the seal resistance by a factor of 10, we would have perfect recordings, and that's what's happening. These are the first recording in 1976. Okay. You see the single channel currents are barely above the noise. After, in 1987, we had improved the seal by a factor of 100, and here you can see single channel currents with a much more improved single noise ratio that allowed us to analyze these currents. And one thing I was interested in at the time is to find out what are the structural determinants of flow through single ion channels. And fortunately, at that time, Shosako Numa, a pioneer in molecular neuroscience, had cloned the first genes, cDNAs, for acetylcholine receptor, and he was kind enough to collaborate with us. So we expressed these channels in oocyte membranes by just transferring the cDNA, or rather RNA, into xenopus oocytes, and then used point mutations to look at the determinants of ion channel flow. And this is a summary of this. I don't want to go into the details. This is a stretch of the amino acid sequence of the acetylcholine receptor called the M2 transmembrane segment, which has a fairly high hydrophobicity index. And we started to systematically point mutate these amino acid at these positions. And what we found out is that the narrow portion of the channel is at the intracellular side of this M2 transmembrane segment, because, as illustrated for the alpha chain, if you replace charged amino acid by uncharged amino acids of different sizes, we can down-regulate the ion channel flow. These are not the only determinants, but it's the determinant that controls the flow of ions through the channel. So, using point mutations, replacing the amino acid at this particular position with amino acids of different side chains, going from glycine to valine, we can down titrate the single channel conductance almost by a factor of 2. So this then gave rise to the following model, which is still true. The wild type channel has a valine, at this M2 transmembrane segment a bulky amino acid, which is a valine. If we replace the amino acids in the wild type by a valine, we get a smaller current. If we replace it by a glycine, we get a larger current. And this is not possible at any other location of the M2 transmembrane segment. So the conclusion from this is that combining single channel current measurements with site-directed mutagenesis in particular parts of the ion channel protein, we have an idea of how the channel is organized. This would be extracellular site, intercellular site. Unfortunately, acetylcholine receptor is still not cloned, but from all we know from the partial crystals or from crystals from other species, this is apparently the valid picture. So to summarize this, we found out how to measure single channel currents, and using point mutation analysis, we could determine some of the important amino acids that determine the properties of this ion channel. Now, the second part, this was all done in the 80s, and I then thought we will go to, I want to see how ion channels cooperate in nerve cells to generate a behavior. And the person who introduced me to this field was Otto Creutzfeldt. By the way, he was a friend of Shentakotai, and we had Shentakotai quite a few times in Munich. Very towering figure. If you ever have you met Shentakotai? And he had very firm ideas how the brain works. But at the time, the data were not all that hard. And Otto Creutzfeldt asked me to, this was before I went into the single channel field, asked me to record and reconstruct neurons from the cat visual cortex. And I tried this for about 2.5 years and ended up with almost nothing that we could publish. And I asked him whether I could do something else. He was very kind. I could do something else that quickly led to a PhD thesis. But after having found the determinants of ion channels, I thought I'll have a go again. This is one of the things in science. You have a failure. As soon as you have a new technique available, you come back to your old problems. And this is what happened in my case. I was always thinking of how I could really accomplish what Otto Creutzfeldt asked me to do, and where I bitterly failed. But then I realized that these PET pipettes that we use for recording single channel currents are very useful. They can be used to record electrical activity from very fine structures like nerve cells in the intact brain. And this is what I'm going to talk about now. So I've been telling you about these ion channels that underlie the two major signals in the nervous system, the action potential and the synaptic potentials. Both of them are due to the opening of multiple of these single ion channels. All the membrane of nerve cells is studded with them. And I was wondering whether we could make some progress in understanding how nerve cells communicate due to the fact that we could use patch pipettes, not only to record single channels, but also to gain access to the soma and to the dendrites in a way that was not possible before, because these patch pipettes or the patch clamp technique allows us to record from very tiny cells up, the tiniest cell that has been recorded is in the order of 3 micrometer across. So I was using this technique to study the interaction of cortical circuits. And if you want to study a cortical circuit, you first must define a behavior that is generated by these particular cortical circuits. You just, if you want to study how the brain works, you cannot start anywhere. You have to have a very defined behavioral paradigm, identify the part of the nervous system that is involved in generating this behavior. Then you have to reconstruct these circuits, and finally you want to measure the signal flow through these circuits in order to understand what is the anatomical and the physiological basis of a particular behavior. Now, the behavior that we chose is shown in this slide. What you see here is a rat trying to decide whether it should jump across this gap, because on the other side there is a chocolate. And the rat does this by using its what's called whiskers. So it uses its whiskers to estimate the distance between the home platform and the target platform. And depending on the information flow that is generated by these whiskers, it decides either to go back or to jump across. So it's a very simple decision, a very simple behavior. It's a binary decision, either to jump or not to jump. And this is what we want to find out. What is going on in the brain of this mouse when the mouse decides to jump, and what's going on when the mouse decides not to jump. We don't know the answer yet, but we have made some progress, which I'm going to show you now. The next slide first shows an actual experiment. So this is a typical experiment. So it palpates, chuck, decided to jump. We have changed the direction, also jumps. Now we are increasing the size. Okay, well, apparently this part of the film hasn't made it. What you are supposed to see is when we increase the gap, the mouse doesn't jump. So a small change of about 1 centimeter is enough to motivate the mouse not to jump. I'm sorry that I couldn't see this, but this is how the experiment goes. Now, what is happening in the brain of a mouse? First of all, we have to find out what are the clues by which the mouse decides on the distance. And fortunately find out that it can make a decision to jump or not to jump across the gap with a single whisker intact. So it doesn't need all the whiskers. We cut away all the whiskers except for one, and the mouse still performs as good as it does with all the whiskers. Now this introduces a very great simplification of our experimental protocol, because we have to find out what's happening in the brain of a mouse when a single whisker is deflected. Now let's just see what the mouse does. This is now a time lapse. Approaches, you see the whisker, whisks, it sort of tries to find out. Okay. Now it has touched once, touches a second time, and so on. After about 6 or 7 of these touches, in this case, the mouse decided to jump. Now, in order to find out what's going on in the brain during this decision, we have 2 behavioral states. First, we must find out what is happening during whisking, when the mouse just palpates. and we have to find out what is happening in the brain when it touches. So these are two behavioral states, and we have to dissect the electrical activity and the cells involved for these two states. And that's what I'm going to show you in the next few slides. The whisker system is particularly nice to work on. It was discovered by Woolsey and van der Loos, and the anatomical determinants are such that a single whisker, when it is deflected, increases the activity of a very small area in the cortex. It's called the vibrissae area or the barrel cortex, and you see a magnification of this here. So when one deflects the D2 whisker, the electrical activity in this D2 column is increased. In order to understand what is going on during these two behavioral states, we first have to reconstruct the anatomy of the D2 whisker or of the D2 column. Now, this was done over the past 6 or 7 years. What we did is we first looked at, is this a laser? Ah, yeah, okay. We first, what we thought is we do a reconstruction of a vibrissae cortex in silico. That is, we register all cells in a column in 3D. We attach the dendrites, which we have been recording from these cells, and we also attach the axons, and then reconstruct a vibrissae cortex in silico. That is, we are not doing this in a single go. We're using different animals to record from different types of cells, register them into a 3D coordinate system, and build a vibrissae cortex in silico. And the result of this, wait a second, okay, is shown here. This is a complete reconstruction of all somata in one particular column. Each of these red dots represents a single soma. If we do a density map, we have to reduce it because you want to do calculations. We get a very clear picture. There is a medium size density, then the density increases in the granular layer, it falls off and then increases again. The density map is shown here. Now we can use this to quantify the distribution of somata in a column. The next thing is to attach dendrites to each of these. We do this by recording from individual cells, reconstruct them, register them, and here you see examples of the 9 cell types, excitatory cell types that we found. They are designated according to the layer in which the soma rests and the shape of the dendritic arbor, and I would like to try to draw your attention to the slender tufted pyramids and to the thick tufted pyramids. These are the 2 types of pyramidal cells that convey cortical activity to the rest of the brain. So most of these cells are used to convey information within the cortex. These 2 types are those cells that convey to the rest of the brain that something has happened. And since we are interested in finding out what's happening during decision making, we concentrated on these 2 types of cells because these cells decide finally whether to jump or not to jump. This is just an example of what I've been telling you. We did a complete reconstruction of the somata of a column. We attached the dendrites. This gives rise to a dendrite column. Since these are about 10 to 15,000 neurons, we are unable at present to register 10,000 dendrites. So what we did is we took a sample of each of these types and then expanded it or extrapolated it in order to reach in order to generate a dendrite column. This, however, is these are all measured data, but for our project where we want to measure the current flows through a piece of cortex, we have to up or extrapolate these cells. But that's not the topic of this lecture. Now, if we look at the electrical activity of these two types of these cells, we find we concentrate on these two cell types that convey information to the rest of the brain. You can see there's a huge difference between the slender tufted pyramids and the thick tufted pyramids. These are very inactive. These are very active. This is quantified in this slide, where you just see the activity following a passive touch by the height of these histograms, and you can see that the major output of the cortex is related to the layer 5 thick tufted cells. I have to say, however, that this is the behavior in the anesthetized animal. We are passively deflecting, and see this huge difference between these two output cells. This is meant to illustrate these two cells again. That's the layer 5B thick tufted cells, slender tufted cells. These are the ones that are very active. These are the ones that barely respond to a touch. So what's going on? Having found that, we said this is passive touch, and this is not what's happening during behavior. You know, we have to find out, or we have to record from these cells in two behavioral conditions. One is during free whisking, and the other one is during touch. So we redid these experiments, which we have been doing in the anesthetized animal, in the awake animal. They are not freely moving, but they are awake, and you will see the experimental arrangement where we can measure the electrical activity of cells during free whisking and during touch. Now, first during free whisking, the first surprise was that in the awake animal, the layer 5 A cells, the slender tufted cells, all of a sudden become rather active. In the anesthetized state, they are almost silent, nothing is happening. If we do these measurements in the awake animal, we find that these cells all of a sudden become very active, and more importantly, the action potentials, which you see here, and the whisking phase have a fixed phase relationship. So this is a trace of the whisker, which we have seen, protraction, retraction, and so on. This is the action potential recording. And if you then ask the question, when do the action potentials occur during the whisking cycle, we find that there is a minimum and a maximum. And in further experiments, which I don't want to go into, we found that this is not the same for each cell. The phase relation between whisking and action potential is distributed over all phase angles. So there is not a fixed relation. What we have is an ensemble of about 1,500 cells, layer 5A cells, that report in their electrical activity different relations of between the whisking and the between the whisking phase and the action potential frequency. Now, how about the other cells, layer 5 cells, that are so active during passive touch? We find that these cells have also increased their activity during whisking, but there is no significant phase relation. So what this means is that these two types of cells report differentially to the rest of the brain what is happening to the whisker. The slender tufted cells report the phase relation of the whisking, and these layer 5B cells, they just increase their frequency slightly, not very much, but there is no phase relation. Now, then you ask, what is happening during touch? That's the second behavioral state. How do these two types of cells react to touch? And the answer is, again, they are very different. Layer 5A cells, the ones that react to whisking, barely show any touch response, as in the anesthetized state, but the layer 5B cells, the ones that respond in the anesthetized state, also respond to active touch. You can see here an experiment. This is the object, that's a single whisker, and we are recording the activity during repeated touches. Each of these gray areas this, corresponds to a touch. This is electrical activity. If you analyze this, you find that before touching, there is an increase in the average frequency, which is not phase related. During touch, there's all of a sudden a major increase in the action potential frequency. Now, this means that apparently the two behavioral states are separately encoded. The whisking state is encoded by layer 5A, slender tufted cells, and the touch is encoded by by the layer 5B cells. Now there is a long debate on how what might be the neural code. And there are two opposing views. One of them maintains that the code, that is, how is some sort of a feature or anything encoded by the frequency of action potentials, it's simply an increase in the number of action potentials. The other view maintains that it is a change in the internal structure of these action potentials. Both ways can be used to convey information. And what we find for this particular cell, or cell type, is that we have both. We have an increase in the frequency upon touch, and we have a change in the internal structure of these action potentials. This is shown in my last slide. If we analyze the internal structure of these action potentials during quiescence, during whisking, and during touch, we find that the number of what we call bursts increases during touch. So not only the number of action potential increases, but also the number of bursts. What you see here is the number of bursts, that is action potentials that occur in triplets, increases significantly upon touch, whereas it doesn't increase during simple whisking. So what it means is, well, I can skip that. That decision-making can be controlled by input from a single whisker. Deflection of a single whisker evokes action potentials in about 4 to 9 columns in a layer-specific and time-dependent way. I just showed you the difference between layer 5A cells and layer 5B cell. Whisking is represented in a column by the action potentials of about 1200 thick tufted cells in layer 5. Layer 5A cells do not report touch. Touch is reported by the activity of the layer 5B cells. These are the thick tufted cells. And it is encoded both by an increase in the number of action potentials and in the internal structure of these action potentials, meaning that the code that is used to convey to the rest of the brain that something has happened, that an object has been touched, must take into account both the increased number of action potentials and the change in the internal structure, that is the increase in bursts. Generally, one can say, based on these experiments, that in two behavioral states, the fact that the whiskers are moving is encoded by one cell type. It's sent to the striatum, and the fact that the whisker has touched is encoded by another cell type, and this is conveyed to the thalamus and to the pontine nuclei. So apparently, the two behavioral states are represented by different cell types, and these cell types report to different parts of the brain. And the question is, where is the decision made? And this is where we are stuck right now. We will have to look into the striatum and into the thalamus to see whether there is a merging of the phase information and of the touch information. And only if we know this, we will be able to find out what is happening during decision making. What are the changes in number of action potentials and the internal structure of action potentials in which part of the brain? My suspicion is it's going to be in the striatum, and find out what are the determinants of decision making. Right now we know that the two behavioral states are nicely separated. They're encoded in the cortex, but the decision is not made in the cortex, it's made somewhere deeper in the brainstem. Now, most of this work was done at the Max Planck Florida Institute, where I spent the last 3 years. If any of the students in the audience are interested in this work, look up www.maxplanckfloridainstitute.org. You'll find part of our work. They have it's a nice area. They have nice grants. And I was collaborating with a number of people whose name I want to mention. This is Marcel Oberländer, Hanno Meyer, Robert Egger, Mike Guest, and Andrew Johnson. And we collaborated with a number of people in Amsterdam, New York, Berlin, and Heidelberg. This is Christian de Kock, who did the freely moving, who did the awake experiments, Randy Bruno, Vince Derksen, and Moritz Helmstetter. All this was done at the Max Planck Institute. You know, our logo, the logo of our society is Minerva. Now, Minerva is not always busy supervising what the scientists are doing. She's also pulling scientists through the waters off the coast of Florida. You can see here a kite. This is Minerva. As Minerva pulls a scientist when we have nothing to do. But sometimes the scientist is not good enough, and this is what happens. So thank you very much for your attention.
Thank you very much for this very nice lecture. And since this was an opening lecture, there is no discussion. So it's, but you might meet Professor Sakmann later. As I understand, Professor Sakmann spends all week in Szegeden, and you will have ample possibilities or chances to talk to him. Thank you very much.
Sakmann professzor úrnak és a levezető elnököknek is. Még egy szolgálati közleménnyel vagyok adós, nevezetesen, hogy nagyon sok szeretettel várunk mindenkit ma este 7:30-kor a Szegedi Nemzeti Színházban, ahol a nyitó gála műsor lesz, és mondhatom, hogy nagyon izgalmasnak ígérkezik a program, úgyhogy javaslom mindenkinek, hogy jöjjön el, utána pedig egy kötetlen állófogadás keretében találkozhatnak a professzor úrral is, meg hát egymással is lehet könnyebben ismerkedni, akkor mindenkit várunk szeretettel este 7:30-kor. Köszönjük szépen, hogy itt voltak.
A projekt az Európai Unió támogatásával, az Európai Szociális Alap társfinanszírozásával valósul meg.